Hi guys, and welcome to our model on muscle tissue. So muscle tissues are among the four tissues of the body. The other three tissues includes the epithelial tissue and connective tissues, which we talk about in a separate model, and then the nerve tissues, which we'll talk about in a later model. The muscle tissues are collections of cells that have the specialized ability to contract, and this contraction makes muscle tissue to be particularly important when producing movement of the body at different parts. So this would include at areas like the skeletal structure, as shown in the image where the man is flexing his arm, and also in organs. Within organs, we have movement occurring. So the movement occurring within organs is made possible by the muscle tissues found within them. So what are the functions of muscles? Muscles have a number of functions that are all pertaining to movement. Number one, they help in producing body movement. So if you're moving from point A to point B, the structures involved in particularly accomplishing that act would be the skeletal muscle. Then we have in stabilizing body position, so just standing, muscles are contracting at that point in time, or sitting down, muscles contract. Depending on the particular posture that you have, standing, sitting, or otherwise, muscles are always involved in there to maintain or stabilize body position. Then we have regulations of organ volume. So this would usually entail muscles found within organs, or we call smooth or cardiac muscles. So these ones are going to be involved in regulating organ volumes, either by contracting or relaxing the muscles to allow for an increase or decrease in the volume of the structures or substances found within those organs. The movement of substances within the body. So if we are moving different substances within different organs from point A to point B, for example, in the gastrointestinal tract, moving food molecules from the mouth down to the anus usually requires muscles along the part of the organs in which this particular substance is being moved across. Then production of heat. Scenarios where we are in a very cold environment, the bodies tend to lead to muscles undergoing spasm, and that spasm is a form of heat production mechanism. So heat production is achieved through the involuntary contraction of skeletal muscles, and this tends to produce what we call shivering. It's that shivering that helps in dissipating the coldness that we feel around us. What are the components or features of a muscle? So we start first of all by the components and features of a muscle cell. Remember that a collection of muscle cells give rise to a muscle tissue. So a muscle cell has a combination of things that tends to be unique to it and distinguishes it from other forms of cells. So first of all, it has a muscle cell. First of all, is known as a myocyte. Myocyte is the ironic for muscle cell or a myofiber, and each of the myocyte has um, particular components that are seen in other cells but have unique names to them. So the plasma membrane of the muscle cell is known as the sarcolemma. The cytoplasm of a muscle cell is known as the sarcoplasma. The endoplasmic reticulum of a muscle cell is known as the sarcoplasmic reticulum. The mitochondria of a muscle cell is known as the sarcosome. Then we have some specialized structures that are not found in other cells, like the T-tubules. T-tubules are specialized invaginations of the sacrolemma that tend to run in a transverse direction across the cell. So they are also known as transverse tubules, hence the name T-tubules. So they're unique to the muscle cells, are not found in any other cell aside the muscle cell. Then we have the transverse systems. Transverse systems are sac-like regions of the sacroplasmic reticulum that tends to act as the calcium storage site. And when talking about the various components of a muscle cell that makes up the muscle tissue, it's extremely important for us to mention the sarcomere. The sarcomeres are regarded as the contractile or functional unit of a muscle. Structurally, they are made up of the structures we mentioned when talking about the um, organelles of the cell. Particularly, I'm referring to the cytoskeleton. Remember, we have different forms of cytoskeleton. So, sarcomeres are repeating units of some of the cytoskeleton, or we call the myofibrils, inside of the muscle cell. They are composed of an intertwined structure of overlapping thick and thin filaments. So the thick and thin filaments in this case would be what we call the actin and myosin filament. So these actin myosin filaments are actually cytoskeletal structures found within muscle cell, and they overlap one another to give rise to sarcomeres. And it's this sarcomeres that would eventually be involved in the contractile function of a muscle. So this is a picture of the sarcomere. You can see that if we zoom into it, you see that it's a collection of um, cytoskeletons, the the filaments, actin and myosin filaments, thick and thin filaments are shown here. So the thick filament, for example, is the one shown in pink here. This is the myosin filament. The thin filament here is shown in blue. This is the actin filament. So how do we classify muscle tissues? We can classify muscle tissues based on two major criteria, based on morphology or based on function. So based on morphology, we classify muscle tissues into either a striated muscle or a non-striated muscle. I'll show you pictures of how that looks like. Then based on function, we classify muscles based on the function either being voluntary in terms of the fact that we can control or we're in control of the movement that those muscles tend to undergo. And then we can also classify into involuntary muscles. These are muscles in which we have no control over in terms of their contractions. Muscles that are found within the organ structures or visceral structures are regarded as involuntary muscles because we can't control how those organs tend to contract or relax. There are three major types of muscles, and using the classification of muscles that we mentioned, voluntary and involuntary classification, and then the striated and non-striated muscles, we'll talk about the different types of muscles in each of those categories. So the three major types of muscles we have include skeletal muscles, cardiac muscles, and smooth muscles. Talk about each of them in the next slide. So skeletal muscles are regarded as striated muscles based on morphology, and they are voluntary muscles based on function. So they are muscles that we can control, and they are muscles that, in terms of their morphology, in terms of how they look like under the microscope, they are striated. The striated aspect of the muscle is just the arrangement of the actin and myosin filaments that we mentioned. And the major features of a skeletal muscle is the fact that it is made up of elongated tubular-like cells. So the myocytes present that makes up the tissue, the skeletal tissue, skeletal muscle tissue, are usually elongated or tubular. And another unique thing is that each of those myocytes tend to contain multiple nuclei. So they have a lot of cell nucleus within the cell, and those nuclei are arranged peripherally. They're not located centrally like other cells. So two things: muscles within the skeletal muscles, the cells within skeletal muscles are striated and voluntary in their function. They contain cells that are elongated and tubular, and then these cells contain multiple nuclei that are arranged peripherally. And the skeletal muscles tend to undergo quick and forceful contractions. So when we want to move from point A to point B, we make use of the skeletal muscles in doing so. The second type of muscle is the cardiac muscle. Cardiac muscle is also striated, but unlike the skeletal muscle, it is involuntary. And the reason is quite simple. It's cardiac muscle because the muscle found within the heart, and we can't control the amount or the nature of contraction in the heart. So the movement or the contraction done by this cardiac muscle is involuntary. However, based on morphology, it's striated in its appearance. The cardiac muscle contains cells that are also tubular, but these cells tend to branch and they tend to unite with other cells around them. So look at a picture like this. Um, and you notice that this is a cell, this is a myocyte here, but that myocyte is not just a singular myocyte by itself. It's connected to other myocytes and it's branching off to form different myocytes. So the branched aspect of the cardiac muscle is what makes it unique and separate from that of the skeletal muscle. Skeletal muscle is just elongated and tubular and well separated from the rest. For cardiac muscles, the cells they are tubular but branched, and that branch, the branch between one particular myocyte and the other in the cardiac muscle tissue, is made possible by what we call an intercalated disc. So intercalated discs are points of union between one myocyte and the other myocyte in the branch in which they form. So this is an intercalated disc. Yeah, these are intercalated discs here that are joining one myocyte to the other. So these are the very important difference between the cardiac muscle and a skeletal muscle. Presence of intercalated discs could make us to suspect that the muscle is a cardiac muscle rather than any other one. Cardiac muscles, the cells they tend to contain about 1 to 2 nuclei. They are not as multinucleated as what you see in the skeletal muscle. And the nuclei tend to be located centrally, not peripherally. They're located centrally. The type of movement or contractions that the cardiac muscles tend to undergo tends to be vigorous but rhythmic contraction. So and the rhythmic contraction there tends to mirror what we see at the end of the day when we talk about the heart beats in a rhythmic fashion, and that rhythmic fashion is by virtue of the cardiac muscle that is found within it that has that vigorous rhythmic contraction to its tone. The last type of muscle we will talk about is the smooth muscle. Smooth muscle is unlike the other two, it is non-striated in its appearance and it is involuntary. It's the muscle that we see in every other visceral structure aside the heart. It contains cells that are spindle-like in shape. This is a typical example of a smooth muscle. This is a smooth muscle cell, spindle in shape, and it possesses some form of intercalated. It is not as mature as what we see in the cardiac muscle, but we do have intercalated discs that tend to unite different small muscle cells to one another. It has just a single nucleus, so if we are to distinguish them, skeletal muscle is multinucleated. Cardiac muscle has about 1 to 2 nuclei present within its cells. What small muscle has just one single nucleus that is located centrally. As you can see, it is a single nucleus located centrally within the small muscle myocyte. And the type of movement or contraction that we see for small muscles tend to be slow. So we have slow contractions that we see in small muscle, and that accounts for the slow peristaltic movement we see in different organ sites that contain smooth muscles. So let's go on down to the skeletal muscle and talk about what we call the skeletal muscle structure. So we'll talk about the skeletal muscle structure in terms of the smallest structure going up to the larger structure. And this just describes for us how each of the different parts of the skeletal muscle are grouped together to form a very large muscle tissue at the end of the day. So we'll start from the smallest to the largest. So usually we start with the myocyte. Myocyte is the cell, the muscle cell, a single muscle cell. When we have a collection of myocytes, as shown here, a collection of myocytes gives us a muscle fiber. So muscle fiber is a collection of myocytes, and these muscle fibers tend to be wrapped around by a connective tissue. The name of the connective tissue that wraps around the muscle fiber is known as the endomysium. So that's the first step. So then we now have a collection of different muscle fibers bundled together and wrapped around by another layer of connective tissue. So you can see in this picture we have different muscle fibers. This one, this two, this three, all of which are wrapped around by a single connective tissue. The name of the connective tissue that wraps around this collection of muscle fibers is known as perimysium. And then that collection of muscle fibers wrapped around by the perimysium is known as a fascicle. So fascicles are collection um, of muscle fibers wrapped around by the perimysium. And then finally, the collection of fascicles, so when we have multiple fascicles now bundled together by a very large connective tissue, we refer to that as the muscle tissue or muscle tissue organ. So the connective tissue that wraps around the collection of fascicles is known as epimysium. So we have epimysium being the connective tissue wrapping around the entire muscle as a whole, the perimysium being the connective tissue wrapping around the collection of muscle fibers or wrapping around to form the fascicle, and then the endomysium, which is the connective tissue that wraps around the muscle fibers individually. So yeah, this describes for us the organization of the muscle of the skeletal muscle as a whole, but also in terms of individual units. And each of those units tend to be supplied specifically by different forms of nerve fibers. So just to describe what I mentioned before, a single muscle fiber tends to be known as a myocyte. It's usually surrounded by an endomysium. A group of muscle fibers will form a fascicle. Fascicle tends to be surrounded by the perimysium. And a group of fascicles will give rise to the muscle tissue, and that is surrounded by an epimysium. So a little bit of deep dive into the skeletal muscle as a whole. So talking about the skeletal muscle and trying to distinguish it in terms of structure to the rest. Skeletal muscle tend to contain well-developed sarcomeres. Remember, sarcomeres are the muscle filaments found within a muscle cell and are involved in the contraction that the muscle cells tend to undergo. The skeletal muscle also has very, very well-developed T tubules and sarcoplasmic reticulum. I mentioned what each of these things tend to represent. And as a result of these well-developed sarcomeres, tubules, and sarcoplasmic reticulum, the skeletal muscle tends to appear like a cross-section of alternating light and dark bands under the microscope. So if you look at this picture, this is a picture of a microscopic view of the skeletal muscle. You notice that there are some dark and light lines shown on the different aspect of the skeletal muscle. So these dark and light lines are not what we call the light and the dark bands. The dark bands are formed by what we are also called the A bands. The light bands are also called the I bands. Each of these things are formed by just the overlapping of those filaments I mentioned. So actin and myosin filaments tend to overlap, and depending on how far or close the overlapping is, they could either form the light band or the dark band, so the A band or the I band. On electron microscopy, the I bands tend to be bisected by a dark transverse Z line, and usually on electron microscopy, then we usually tend to see that the sarcomere tends to extend from one point of the Z line to the other point of the Z line. That's a little bit too complicated for you to understand, but especially when you get to the aspect of physiology on nerve and muscle, I think it will make a lot of sense. In terms of this particular model, this is an introductory model. What you just need to get is the fact that first of all, the skeletal muscle has well-developed sarcomeres, titobos, and sarcoplasmic reticulums. And the sarcomere tend to have different thin and thick filaments, myosin and actin filaments that intertwine together. That intertwining gives rise to the appearance of light and dark bands, which is what we can see. At least we can see the light and dark bands, those lines, those striated lines. Those dark and light bands is what makes us say that a skeletal muscle is striated in the first place, because you can see that it forms like stripes, and the dark bands are called I bands. The dark bands are called A bands, rather. The light bands are called I bands. Yeah, this image depicts for us those striations and the different zones I mentioned. So you can see that the actin and myosin filaments um, arranged would give rise to the sarcomeres, and you can see how they overlap to form different aspects here. You can see that, for example, the overlapping between the actin and myosin filaments, as you can see, where they are both on the same level, would give rise to a darker band. And then where we just have actin filaments alone, that gives rise to the light band. So the light band is the band that contains just actin filaments, thin filaments, and the reason why it is light. The dark band is when we have a combination of the thick and thin filaments in on one lane, and that's what gives rise to the dark bands. And each of these different bands have some zones to it. Now, for example, the H zone is the middle of the thick filament, is the middle of the A band, or the middle of the dark zone, which just contains the thick filament, the myosin filament. Then we have the Z line. Z line is just the beginning of one sarcomere to the end. So Z line marks the start of a sarcomere and the end of another sarcomere. And this is Z line here. So in between the Z line is the entire sarcomere. So it contains the I band at the edge, and then the A band in the center, and then the H zone at the center of the. So like I said, this is a little bit highfalutin. Once we get to it, the aspect of physiology that deals with this, we'll go into it in depth. But in terms of just understanding it, I think it's fine just knowing it in terms of the component of the sarcomere, and then the arrangement of the different filaments within the sarcomere. Let's talk about the structure of the cardiac muscle. Cardiac muscle, in contrast to the skeletal muscle, has T tubules present within it. The T tubules of the cardiac muscles are large, but they are larger in the ventricular muscles compared to the other aspects of the heart. The cardiac muscles do not have a well-developed sarcoplasmic reticulum, and they tend to have different. They give a form of appearance, tissue appearance of chains, as shown in the image. Remember that the cells of the cardiac muscles tend to be branched, and they are connected by intercalated discs, and as a result, they tend to appear like chains rather than appearing in a well organized striated form or parallel striated form. So cells within the cardiac muscles tend to each have bifurcations or branches I mentioned, and in terms of it, the tissue appearance of the cardiac muscles, these branches that the cells tend to form gives it an appearance of what we call a syncytial network. This is what I mean. So you can see this is one cell here, it's a branch to form another cell here. That one branches to form different other cell here. So you can see the branches that are formed by each of the cells gives rise to this syncytium, or what we call a network, that is made possible by what I call the intercalated disc. I mentioned that already. So the cardiac muscles also have their form of T-tubule and sarcomere, and they have what they call a Z band rather than an A to I junction. Um, uh, that I also would say is also highfalutin. So once we get to the aspect of physiology that talks about that, we'll dive into it at a deeper level. But in terms of just understanding what the cardiac muscle have, it's just important to note that the tissue appearance of the cardiac muscle gives rise to a syncytium network look or appearance, and this is by virtue of the fact that the cells tend to branch, and then the branches tend to be connected by what we call intercalated. And finally, the smooth muscles in terms of its structure lacks a sarcomere, and that's the reason why it is not striated. Remember that smooth muscles are non-striated and involuntary. The reason why they are non-striated is that the sarcomere appearance that we talked about for cardiac and skeletal muscles is lacking in the smooth muscle. T-tubes are also not present, and then the sacroplasmic reticulum is also rudimentary. You can see that in terms of the components that we saw for cardiac and smooth muscles, they usually or virtually lacking totally in the smooth muscle. So the thick and thin filaments do exist within the smooth muscle, and the sliding of the thick and thin filaments tend to generate what we call tension or contraction at the end of the day. However, these thick and thin filaments are not organized to form the sarcomeres as mentioned, and the form of contraction that we tend to see that makes it to be slow tends to be a twisting motion or a twisting contraction, and as a result that twisting contraction is what tends to allow slow contraction occurring within this muscle, contrast to what we see in the cardiac muscle and in the skeletal muscle, whereby we have a much forceful and vigorous contraction. And that concludes our module on muscle tissue.